Okay, hello and welcome to Biology Class 1. Today we'll be covering molecular biology. My name is Destiny and I'm joined by my co-tutor, Malaz. A little bit about Pre-Med Peers. We are a nonprofit organization and all sessions will be recorded and posted after class. Today we'll be going through content related to molecular biology, followed by reviewing the practice homework passage, then a few practice questions. Please ask if you have any questions at any point in chat or by unmuting, and I will also link the passage for everyone for later. Nucleic acids, what are they? Well, first, they're macromolecules formed of nucleotides and they carry genetic information, allowing it to be passed on. This includes DNA and RNA, as well as some synthetic versions that we don't really concern ourselves with. Nucleotides, the building blocks of nucleic acids, consist of a sugar, phosphate, and a base, while nucleosides are just the base and sugar. The phosphate group is what allows for the formation of the nucleic acid, as polymerization relies on formation of a phosphodiester bond, which involves phosphate groups and hydroxyl groups. This linking of nucleotides is a condensation reaction, and the mechanism is shown here. Does anyone have any questions?
I do have a question I want to ask everyone. Do you know why they call it condensation reaction?
It's because you release a molecule of water
That is
once
correct.
you join the phosphate
Yes.
and hydroxyl group.
Right. Awesome. So the genome refers to all of the genetic content of any cell. Prokaryotic genomes are often smaller than eukaryotic ones. Additionally, genomes of eukaryotic organisms are found in the nucleus, while prokaryotes' genomes may be found in the nucleoid region, which is technically in the cytoplasm. Prokaryotic chromosomes are often circular, while eukaryotic chromosomes are linear structures. Also, the central dogma refers to the process by which DNA leads to some product or by which cellular information flows in the cell. It can best be summarized as DNA to RNA to protein. Any questions about that? Awesome. So mutations or alterations in the genome's nucleotide sequence can happen in various ways. First, the polymerase enzymes involved in DNA replication and repair can sometimes make mistakes and insert the wrong nucleotide, additional nucleotides, or not enough nucleotides into the sequence. Mutations can also be the result of damage. Damage from external agents such as radiation is called exogenous damage. Damage that results from normal metabolic processes is referred to as endogenous damage. Mutations may also result from transposition, in which some section of DNA is moved from one DNA molecule to another. Such a section is called a transposon or transposable element, and as you can imagine, these can easily disrupt genetic sequences and even significantly change the genome size. There are also many ways of categorizing the types of mutations that can occur. One subject often found on the MCAT is point mutations. Point mutations involve altering only a single nucleotide and can refer to substitutions, insertions, or deletions. Based on its effect, these point mutations can then be placed into other categories: silent, missense, or nonsense. A silent mutation has no effect on the protein product. A missense mutation changes an amino acid in the protein product. A nonsense mutation changes an amino acid codon into a stop codon. Mutations may also be much larger, as in chromosomal mutations, which change chromosome number or size. Mutations of this sort include deletions, duplications, inversions, insertions, and translocations. Does anyone have any questions about this or anything related to this? Alrighty. While mistakes are inevitable, these errors are often repaired during or after replication. During replication, the DNA polymerase is proofreading the bases it is adding. If there is an incorrect addition, DNA polymerase will use its exonuclease action to release the incorrect nucleotide, then it will add the correct one. Other corrections occur after replication. For example, if a small incorrect insertion or deletion occurs, a pair of complexes recognizes this error, binds to the problematic bases, and cuts the DNA around the mismatch. Other enzymes will then cut out the section of DNA containing the mismatch, and DNA polymerase will insert the correct sequence of nucleotides. Then DNA ligase will seal the DNA cut. DNA damage, such as from radiation, may be chemically reversed, and damage from chemical reactions may be undone by certain enzymes in a process called direct reversal. Additionally, if the double helix becomes distorted due to a few damaged nucleotides, a helicase will unwind the DNA so that the problem area, as well as a surrounding patch, can be removed. DNA polymerase once again then fills in the sequence, and DNA ligase seals the backbone. This is called nucleotide excision repair, and note that it involves a cut at both the 3' and 5' ends.
I have a question.
Yes.
So for nucleotide excision repair, is this only in respect to like exogenous mutations? Because I see the first step says UV radiation. So does nucleotide excision repair also occur if like something just goes wrong in the cell, regardless of the external environment?
It's usually a result of radiation.
Okay,
I think
thank you.
there's usually a nucleotide excision repair, unless it's for radiation, and it's often that thymine, because the radiation causes them to undergo some reaction that allows them to make dimers. So that's the real cytosine too sometimes, but particularly that thymine is the main thing I see, even in terms of questions. But I
Okay,
don't
thank you.
think I've ever seen it not be radiation.
And one reason I would like to add why usually it's thymine, because if you were to think about it, you're removing a whole nucleotide there. You're actually removing a whole segment of the DNA. And the reason why, because in the thymine dimers over here, you cannot reverse the damage by taking out one pair. You have to take out two of them or the whole strand to fix it. So this is why it happens mostly with radiation, because it causes a lot of thymine dimers.
Double-stranded breaks are especially dangerous as it can lead to loss of large segments of DNA. This issue can be resolved through non-homologous end joining or homologous recombination. In the first, the break is resealed, often with the addition or removal of a few base pairs. This method is referred to as messy because it results in mutation necessarily. but it is still an improvement over loss of a huge chromosome segment. The other method, homologous recombination, copies information from a homologous chromosome and uses it to repair the break. I know one question I had about this is like, why ever do this first method when it's so messy? So I looked that up, and it turns out it just depends on where in the cell cycle you are and what's going on around and what proteins are available. Because sometimes you can't do the non-messy method simply because the proteins necessary for it aren't around. Okay, I found this video helpful. Sometimes it helps to see the process, and I'll cover it and summarize it afterwards.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping.
I think someone said there's an echo. It helps if everyone is muted, and I will also mute myself.
Is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Based on this, I just want to ask everyone a question, because I believe this is an important concept. Does everyone know what direction is DNA read and synthesized? Uh,
5' to 3'.
which one? Is that the reading or the synthesizing?
Reading.
Reading actually the opposite.
Huh.
DNA is read 3 to 5. So if you're to think about it, you don't read a book from the end to the beginning. You read the book from beginning to end, right? So reading is always 3 to 5. But synthesis is the opposite. So you synthesize from 5 to 3. The MCAT tend to trip you on this topic a lot. They type this different ways, but it's very important to know that the synthesis is 5 to 3 and the reading beginning to end or 3 to 5.
That is a very important concept, and I would say that's relatively high yield as I've seen it on multiple practice exams. And in that vein, just to summarize it, when we're talking about DNA replication, you definitely want to remember that it's semi-conservative. They'll ask questions based on this idea. It's only in the 5' to 3' direction. And on one strand, it will be continuous, and on the other, it will be discontinuous. Also, does anyone have any questions on this topic? Okay. As you can see in the images, prokaryotic and eukaryotic replication are a bit different. First, the prokaryotic DNA is circular, while the eukaryotic DNA is linear. Due to this, only eukaryotes have the protective telomeres and telomerases. Additionally, there will be multiple origins of replication on each eukaryotic chromosome, while there is only one for each prokaryotic chromosome. Different DNA polymerases are primarily used for prokaryotes versus eukaryotes. The rate of replication is also much higher for prokaryotes. However, both use helicase to unwind the DNA. Both involve replication forks. In both, replication will proceed in a 5' to 3' direction, and both have that discontinuous lagging strand and the continuous leading strand.
I just want to unmute and say a couple things. Caitlin, response to your question, I am not sure what is the reason why it's 3 to 5, but anti-parallelism could be related somehow, but I'm not sure how. The other thing I was going to say, Justin, if you can go back to the previous slide, I just want to ask another question because it's
Yes.
quite important. Does everyone know what is the difference between the templates and the coding strand? Because I also believe this is very important.
So the template strand is the sense strand, and the coding strand, or it's the other way around, I believe, actually. No, template strand is sense strand, and coding strand is antisense strand. So it depends on which strand you're reading and replicating from.
So the coding strand actually looks like the mRNA that is synthesized, except you have the thymine and uracil. And the template strand is the strand that you make RNA from. So they might ask you like questions based on which is sense, which is antisense, which is template, which is coding. So it's important to know the differences between the two and be able to differentiate them based on their transcription.
It's very true. Very true. Oops. Okay, So as said before, DNA and RNA are both nucleic acids used to carry information consisting of nucleotides. However, as you can see, DNA contains the sugar deoxyribose, which lacks a hydroxyl group on the second carbon. RNA contains the sugar ribose, which has this hydroxyl group. Additionally, DNA is double-stranded, while we typically see RNA in a single-stranded form. This isn't always the case, but this is usually the case. Cell-based life, eukaryotic or prokaryotic, uses DNA. However, viruses may use RNA or DNA for their genomes. Also, in viruses, sometimes they use double-stranded RNA. RNA plays many important roles in the cell and comes in many forms. Some RNA serves a coding purpose, such as messenger RNA, while other RNAs are non-coding. Messenger RNA is protein coding. Ribosomal RNA and transfer RNA are non-coding but essential for protein synthesis. Other non-coding RNAs of importance include microRNAs and small interfering RNAs, both of which play regulatory roles in the cell. We also have some other RNAs where we're not quite sure what their purpose is quite yet, or if they even have one. Any questions there?
One thing I would like to add that the small nuclear RNAs, especially like the silencing RNAs, they usually tend to regulate translation by inhibiting it, by binding to the messenger RNA. They tend to ask us in passages, and it's usually to silence translation.
Yeah.
There is also the shRNA or small hairpin RNA that also is involved in here that does similar things.
Transcription refers to the process of making an RNA strand from a DNA template. This RNA strand is built by RNA polymerase through base pairing with the template DNA strand. The resulting RNA strand will be almost identical to the non-template DNA strand, except there will be uracil nucleotides instead of thymine. The process of transcription can be divided into initiation, elongation, and termination. First, the DNA double helix is split into single strands. Then, RNA polymerase binds to the promoter in the initiation phase. RNA polymerase then proceeds down the template strand in the 3' to 5' direction, creating the pre-mRNA transcript in a process called elongation. Finally, the RNA polymerase will reach the terminator DNA sequence. The resultant mRNA molecule will eventually be used to create protein. Thus, transcription is the first step in the central dogma. Any questions there? Okay. In translation, the mRNA created in the previous step is read to determine what amino acids the polypeptide will consist of. The nucleotides of the mRNA are read in groups of 3 called codons. The start codon, AUG, codes for methionine and signals the start of polypeptide synthesis. Similar to transcription, translation has those same 3 phases, which is the initiation, elongation, and termination. Transfer RNAs link mRNA codons with the associated amino acids through use of the tRNA's anticodon. tRNA is a strand of RNA arranged into a folded structure, often called the 3-leaf clover, with an anticodon on one end and an attachment site for amino acids on the other. The tRNA is loaded with its amino acid by amino acyl tRNA synthetases. While tRNA can be thought of as a bridge, the ribosomes are often thought of as the polypeptide factories. Ribosomes are where the polypeptides will be built. They consist of protein plus ribosomal RNA. The large and small subunits of the ribosome, as well as the mRNA of import, come together during initiation, and the ribosome becomes the translation site, as well as catalyzing the reaction that links the amino acids in the polypeptide. The ribosome, mRNA, and tRNA with the start codon are called the initiation complex. In elongation, peptides are formed between amino acids, and the mRNA is pulled along through the ribosome with new codons and their tRNA in the A site, previous amino acids in the P site, and empty tRNAs exiting through the E site. I think that one's easier to remember because you can just look at the first letter. So you'll have an amino acid in A, the polypeptide in P, and E for exit. When translation is complete, coded for by stop codons, the subunits will come apart, releasing the polypeptide, and the ribosome can be reused. This is termination. Any questions? I think on this, one thing that jumps out at me is that if you have an insertion or deletion, if you look here, you can see how it could change everything downstream because it changes the reading frame. So that's one thing to keep in mind. And when we're talking about point mutations, which often comes up on the MCAT, you would prefer a substitution over an insertion or deletion for exactly that reason.
One other thing I would like to mention that when knowing what type of mutation it is, it's also helpful to know the stop codons because you could just change one base pair, but that could make something a stop codon and you have a short protein that could be non-functional. So it is also important to know the stop codons, what they are, so that you would know if you change something, if there is a stop codon as a result of this change, you can have a non-functional protein as a result. They tend to ask good amount about that sometimes.
That's true. And also know that sometimes you can have a substitution that does not change the amino acid because we have what are called, it's wobble pairing. So at that last base, it can change a little bit and still result in the same amino acid.
So in other words, wobble pairing is one amino acid is coded by different codons, or different codons code for one amino acid. So that if you have a change in any one of them, you can still code for that same amino acid.
Assuming it's in that last base, exactly. That's a good way to phrase it. Okay.
And since we talked about transcription and translation, how they're done, it's also important to consider how these two processes are regulated. These two processes can be regulated when they are being done on the spot, and they can also be regulated after they are done. So for the transcription, usually we touched a little bit about the concepts of promoters, which are regions of the gene that the polymerase can bind on, but also other transcription factors that could enhance or repress the protein transcription. So this is when you can regulate the transcription when you are doing it on the spot, but you could also control transcription after it's done. And this is usually done when you control the mRNA. And this could be done by alternative splicing, or you can degrade the mRNA. Also, one thing you can do is you can control how the mRNA is translated by controlling how fast it is usually done so. You can also do it after it is translated. But generally, the MCAT focuses a lot about the first two, meaning that what factors bind to the DNA, how do they affect the transcription, and also what happens if you change the mRNA. Usually that could lead to degradation of the mRNA, hence you have no protein production, or you could change up the mRNA by alternative splicing so you could have new unique proteins. So the first two usually are what's being asked about in the MCAT, at least from my experience. And when we compare the mRNA, the messenger RNA, between prokaryotes and eukaryotes, there is an important fundamental difference to know between those two. So we talked about mRNA is translated and is coding for a protein, but there's a difference where the eukaryotic mRNA, we call it monocistronic, hence the name mono comes from. Basically, one gene codes for one protein. But in the prokaryotic mRNA, it's called polycistronic, where one operon, basically a segment of DNA that is controlled by a promoter. So one promoter controls the expression of multiple genes, or I'm sorry, multiple proteins. So basically, one mRNA could be translated into different proteins or multiple proteins, whereas in eukaryotes, one mRNA codes for one protein. They tend to ask about that usually, so I thought I would bring it up. Do you have any questions thus far?
I have a question.
Yes.
So you're saying that prokaryotic mRNA is polycistronic, meaning that one mRNA can code for multiple proteins. So my question comes with, like, I guess this is post-transcriptional or whatever, pre-transcriptional modification. So is alternative splicing only the removal of introns? It doesn't affect like how many proteins are going to be created from that mRNA?
Usually, alternative splicing takes place in eukaryotes, and usually it does not affect codons. Basically, how you cut off the introns will result in different mRNAs. So, based on my understanding and based on what I know, that it does not affect the codons. It's mostly how you cut off the introns affects what is the resulting mRNA. And it usually happens in eukaryotes. Does that answer your question?
Yes, it does. Thank you.
Of course. Does anyone have any other questions? Okay, we can move on then.
Okay, just to wrap up the content review. So after translation, it is folded into its proper structure, often assisted by chaperone proteins. However, it may then be cleaved or even have various functional or chemical groups added to it. So after translation, that's not necessarily the full product. We still have some alterations to do, and I've added this to summarize some of the possibilities.
One
Yes.
comment I would like to say, usually for phosphorylation, it happens with serine. Whenever they ask about phosphorylation, they usually are referring to which amino acid is serine. At least this is what I saw.
It can also be any of them with a hydroxyl group
Correct,
that can
yes.
be phosphorylated. So you might see a threonine and what's another one? Phenylalanine, I believe. No, tyrosine.
Yes, tyrosine, threonine, and serine. But a
Yeah.
lot of the questions, they focus on serine for some reason, at least this is what I saw. But generally, in order to do phosphorylation, having that hydroxyl group is important. And knowing which amino acids contain those this functional group is important to know what type of post-translational modification the protein can have.
That's important to build those associations. And on that topic, I would say for acetylation, I think lysine, because that's what I usually see. And for the disulfide bond, that's pretty much only cysteine usually. Would you
They
agree?
didn't, yeah, they ask a lot about cysteine disulfide bonds, yeah.
Okay, that concludes the content review. Next, we'll move on to reviewing the passage. Did everyone complete it, or do some people need a moment?
That's the homework, correct?
Yes.
Okay.
It was called The Fundamental Questions, DNA Replication 2.
Yeah, I completed it.
Awesome. Okay, we'll jump right into it and review it. So did anyone have any problems with question 1?
No, this one was easy.
Okay. What did you get for number 1?
Being copy would be replication. So B.
Right. Was this one easy for everyone? Okay. I'll keep going then.
Do you want us to answer?
You don't necessarily have to. I think I'll just focus on trying to take the temperature regarding it. For example, I think this one was probably relatively easy. Am I right?
Mm-hmm.
And is everyone familiar with the experiment that determined this?
No, I forgot, to be honest.
In the slides, I included a link summarizing the experiment that determined whether it was semi-conservative or not. It was pretty interesting. They basically looked at so they altered the density of the nitrogens on the original strand, and then they let it replicate, and they looked at how each differed. And I'll include a link to that in the chat as well. It's also in the PowerPoint. Next to the slide where I summarized that it's semi-conservative, I included a link to the experiment that determined it, and it's pretty interesting.
Also important to understand that experiments because they tend to give you passages similar to that experiment and they ask you questions about it. So if you understand it, you can answer similar questions about it.
Yes, and it allows you to go a little bit quicker sometimes as well, because they'll often go ahead and go through it, but it can be confusing if that's your first run through. And here's the experiment. I linked it in the chat, and it's also on slide number, the one that says DNA replication is. Okay. Can anyone answer this question?
C.
Helicase.
Exactly. So when you think unwinding, think helicase. And when you think sealing, which enzyme do you think of? Exactly, ligase. That wasn't asked, I just wanted to check. I thought this one, did everyone agree this one was pretty easy? Yes, exactly. You've got to unwind it before you can do any replication. What did you guys get for this one? Oh, it was a question. Look at me. So we covered this one. The function of DNA ligase is sealing those nicks. And that relates to combining the fragments. A whole lot of C's in this one. Exactly. If you see those Greek characters, you should be thinking eukaryotic, definitely. Okay, awesome job, everyone. I will move on to the practice questions now.
All right. So for this question, it is asking about adjacent bases within a linear strand of DNA. So it's important to highlight the word within a strand of DNA. And the options we have are hydrogen bonds, ionic bonds, covalent bonds, and phosphodiester bonds. When you see the words within a DNA, that's usually mean this has to be connected via a covalent bond, because you have to maintain that strand of DNA cohesive, right? You cannot have hydrogen bonds or ionic bonds because these bonds are not as strong enough. And phosphodiester bonds are a form of covalent bonds, so that makes the answer D correct in this case. Does anyone have any questions about this one? All right, I guess we can move on. It seems like people are responding quick to that one. So you see the difference here? Here it's talking about between two strands of DNA. In this case, yes, it is hydrogen bonds. But when you think about within a strand of DNA, it's phosphodiester bond, because you talked about to unwind DNA to replicate it, transcribe it. So basically, you cannot have covalent bonds because you cannot take them apart. Hydrogen bonds are both stable enough and at the same time are not as strong, so you can break the binding of them and unwind the DNA. So it's important to know what bonds connect adjacent bases, both within a strand and between strands.
It could also be because when you have those hydrogen bonds, for each base pair, there's multiple hydrogen bonds. I think for A and T, it's one of them it's 2, and for the other it's 3.
Oh, they always love to ask about melting points.
Oh yes.
For those. So it's whenever they ask you which DNA strand is more stable, which DNA strand has a higher melting point, or basically which one is harder to break, the one that has more C's and G's, because these can form 3 hydrogen bonds, and hence they can make stronger bonding between the 2 strands of DNA. This concept is always being asked, like almost on every practice exam. So
They
it's
love
good to
that
know.
one.
Yep.
Is it higher melting point for C and G?
Yes.
Yes. Yeah.
Okay.
It is harder to break. Seems like we're getting a lot of B's for this one, which is the correct answer. But I had a question I wanted to ask. Does anyone know the difference between an operator site and a promoter site?
Well, I do know that in prokaryotes that have operons, the repressors or the activators would bind to the operators.
Yes, that is correct. So the transcription factors that could be both, they could enhance transcription or repress transcription, hence activator and repressors respectively. These combine the operator site. Also, some transcription factors could bind the promoter site. So a fundamental difference between these two is where do DNA polymerase bind? It only binds to the promoter site. Transcription factors could bind either, but RNA polymerase only binds to the promoter site. The other question that I wanted to ask is, does anyone know the difference between an operon and an operator? Well, I just described operator, but the difference between operon and a promoter then. I can go ahead for that. So they also tend to ask about that a lot. Operon is basically a segment of DNA that is controlled by a promoter. So an operon contains both the promoter and the gene. They tend to ask a lot about these terms, make sure you differentiate them well. So again, the operon is a segment of DNA that is that contains a promoter in a gene.
Some I have that same rule. When in doubt.
Actually, the answer for this one is A. Can you go back to the slide that shows the bases?
Yes.
When in doubt, go with A. Yeah, you can do that if you want. Could you present? Yes. So here's the thing. You do not have to know how to draw these. You only have to know how to differentiate them from each other. So for example, if they ask you what is the difference between an adenine and a thymine, well, we know that one of them is a purine, one of them is a pyrimidine, so it's easy to do that. But within each group, it tends to be harder, right? Because guanine and adenine, both of them are purines, but the difference between them is, you can see here, is the carboxylation pattern, where guanine, the only one that contains a carboxyl group. But if you look at an adenine, it does not contain a carboxyl group. So it's good to know. Also, it's important to know the differences between the pyrimidines, where that question didn't ask about them. But just the differences, I mean, is basically the functional groups. Pure as gold. Yeah, it's a good mnemonic. And also, pyrimidine is CUT, cut pyramid. I think about it that way. With cytosine, uracil, and thymine cuts. But it's important to know the differences between within each purine or pyrimidine group based on the functional group. I think of cutting the pie. Yeah, that works. Actually, that works nice. Yeah. But yeah, does anyone so far have any questions?
Can you repeat the pure as gold?
Sure.
Again.
Sure. Could you present it again? Sure. So basically, as is A, adenine, gold is guanine, or G. Purine is pure. So pure, purine, as adenine, gold, guanine. Does that make sense?
Yeah. I just didn't know what it was like standing for, like the letters, but got it. Yeah. Thank
Oh yeah,
you.
of course. Mnemonics are a lifesaver on the MCAT. Any other questions?
Hi, I just hopped on. Did this meeting start before 8?
Yes.
Oh,
Started around
okay.
7, yeah.
Oh, it's 7?
Yeah,
Okay, thank you.
of course.
However, it will be recorded. I have it recording now, and I will post it on YouTube as soon as I'm able.
Thank you.
Actually, Destiny, could you go to the 5th question that I moved along? I think this is an important question also. They ask about that a lot. Yeah, so Yes. Yes. This is the reason why I included this question, Caitlin, because it's important to know, yes, there is a methylation difference between them, but it's also important to know which one has it. So in this case, thymine contains a methyl group, if you were to present this again. And uracil does not have a methyl group. So in this case, the lack of methyl group on uracil, it's what makes it different. So the answer B is correct. Does that make sense? I'll take that as a yes. The MCAT loves functional groups, so make sure you know them.
Just want to thank you all for being here. Once again, it will be recorded and posted. Later, the slides will also be available in Google Classroom. They actually already should be. And feel free to email us with any questions. We're also in the Slack group, am I right? So I know I'm definitely open to getting any messages from there, however you need, and let us know.
Same here as well. I'm also available via Slack, so feel free to shoot me a message if you have any question.
Thank you.
Thank you.
Have a good night.
Thank you.